Hi friends, today we are going to see the past year NEET questions for the chapter Cell Cycle and Cell Division. Today in this video we will cover part 1. Now let's begin with the first question here. DNA replication in bacteria occurs dash. So this question was asked in the year 2017. Now coming to the solution. Now you're given 4 options here: within nucleolus, prior to fission, just before transcription, during S phase. Now before we go directly into the answer, what is DNA replication first of all? We're talking about the bacteria here. So in bacteria, the initiation of replication occurs at the origin of replication. We all know that origin of replication. Okay. So what happens here? The supercoiled DNA will unwound itself, right? It will unwound itself with the help of the enzyme called as DNA gyrase. We have many enzymes like polymerase, topoisomerase, and many enzymes. So DNA gyrase will help the DNA to get unwound. Okay. So what happens here? The made single-stranded by helicase, and they are bound by a single-stranded binding protein to maintain its single-stranded state. So DNA replication in bacteria occurs prior to fission. Okay, the correct answer is option B, prior to fission. In bacteria, the DNA replication occurs in the cytoplasm. We all know that it occurs in the cytoplasm prior to fission. Prokaryotes, due to the primitive nature, they do not show a well-defined S phase. What is the other name for S phase? Synthesis phase. Therefore, the correct answer is option B, prior to fission. To your right side, I have given you the common DNA replication process that takes place in eukaryotes, where there are presence of different enzymes like DNA polymerase, then you can see the topoisomerase, the lagging strand, the leading strand, the gap, the helicase enzyme, the RNA primer. So these are some of the important components when it comes to DNA replication. Therefore, the correct answer is option B, prior to fission. Now let's move to the next question here. This question was asked in the year 2015. Okay, a somatic cell that has just completed the S phase of its life cycle as compared to gamete of the same species has dash. Again, this is an easy question, but only thing you need to remember the amount. See, the somatic cell will undergo, it will have a diploid state, so it will be 2n. So twice the number of chromosomes is for sure. Only thing you need to know here, is it 4 times the amount of DNA or twice the amount of DNA? So if at all a confusion comes, it will only come in this part. We all know it is twice the amount, twice the number of chromosomes. We all know it is 2n. But only thing we need to understand whether the DNA is 4 times or twice the amount. The correct answer is option B, twice the number of chromosomes and 4 times the amount of DNA. Okay, therefore the correct answer is option B. When S phase completes their somatic cell, see, once the S phase completes, the somatic cell will has 2 number of chromosomes, that is 2n number of chromosomes and 4c content of DNA. Therefore the correct answer is option B. Okay, next question. This question was asked in the year 2014. During which phases of the cell cycle amount of DNA in the cell remains at the 4c level if the initial amount is denoted as 2c? Okay, you're given 4 options here. You have G phase, they are talking about the G1, G2, G0 or G null phase or M phase. You have S phase. So what are these phases actually? Why is it important in the cell cycle? Each phase has a different function to carry out. Now if you take the S phase, we're talking about the S phase now. The S phase is a phase of the life cycle, it's a phase of the cell cycle in which the DNA is replicated, which occurs between G1 and G2. Right. Since accurate duplication of the genome is a very critical part of the human cell or any eukaryotic or prokaryotic cell, this S phase, if you see, is actually tightly regulated and widely conserved or widely monitored phase of the whole cell cycle. So S phase I've explained. Now what about the G phase? How can you explain G phase? What is the activity there? Now G phase is composed of G0, G1 and G2. Now the G phase describes the cellular state outside the replicative cell cycle. Classically, cells were thought to enter the G0 primarily due to the environmental factors like nutrient deprivation that limited the resources necessary for the proliferation. Now coming back to the question, I've explained to you what is S phase, G phase and so on. Now here they are asking us the initial amount is 2C, right? Okay, is denoted by 2C. And what if at all the cell remains at 4C level if the initial amount is denoted as 2C? This kind of thing is actually seen only in G2 phase. Okay, it is not seen in any other phase. In M phase, both 4C and 2C of DNA are present in the different stages, but when we talk about 4C and 2C, it will come only to the G2 phase. Okay, therefore the correct answer is option C. And to your right side you can see the different phases. G1 is responsible for growth, S phase it is responsible for DNA synthesis, G2 it is responsible for the growth and preparation for mitosis, and M is responsible for the mitosis, which is cell division. Okay, therefore the correct answer is option C. Now next question. This question was asked in the year 2014. Okay, in S phase of the in S phase of the cell cycle dash. Okay, so already in the earlier slide we discussed what an S phase is, right? What is an S phase? How can you describe it? S phase of the cell cycle in which the DNA is replicated and it occurs between G1 and G2 phase, right? Since accurate duplication of the genome is a critical process, we also saw that this S phase is tightly monitored and highly or widely conserved process. Now in S phase, what happens here? The amount of DNA, does it remain as such, or does it double? What is the answer? The correct answer to this question is option A. Amount of DNA doubles in each cell. During the S or the synthesis phase, the replication or the duplication, you can either call it as replication or duplication, okay, so when we talk about replication and duplication, we are talking about the chromosomal DNA. So during the S or the synthesis phase, the replication of the chromosomal DNA, which happens to occur with the help of which protein? Any idea? We always have the help of the histone proteins. They take place, and during this time the amount of DNA will actually double per cell. Therefore the correct answer is option A. Amount of DNA doubles in each cell. Now moving on to the next question. Look at this question. You have a diagram here. Given below is a systematic breakup of the phases, stages of the cycle. So you have the alphabet and the function what they represent. Now if you see A, they have mentioned it is cytokinesis, B they have mentioned metaphase, D synthetic phase, C is karyokinesis. Out of which one will actually match the function of the given stages? Now the correct answer is option B, okay, D. So where is D? You have the D here. So it represents the synthetic phase. So this is right. Which one of the following is the correct indication of the stage or the phase in the cell cycle? So this is actually the correct indication where we talk about the synthetic phase. Okay. So here they have clearly mentioned in systematic breakup of the phases, that is, or you can call the stages of the cell cycle. D, the synthetic phase, is the correct indication of this stage. See, all this actually represent the bad indication or the wrong indication. Only D represents a correct indication. Therefore, option B, D, synthetic phase, is the right answer. Okay, let's move on to the next question here. This question was asked in the year 2005. At what stage of the cell cycle are histone proteins synthesized in eukaryotic cells? Okay. So you're given 4 options here: during G2 stage of prophase, or during S phase, or during the entire prophase, or during telophase. Okay. We already saw in the earlier slide the protein that is responsible, or which plays a major role in chromosomal DNA replication, is histone protein. Okay. Histone proteins are synthesized during the S phase of the cell cycle, and we also know that S phase is also called as synthesis phase. S phase or the synthetic phase is when the synthesis of DNA takes place. Therefore, the correct answer is during the S phase. At what stage the cell cycle are histone proteins synthesized in a eukaryotic cell is during the S phase. Okay, S phase is the first phase that it undergoes. We need histone proteins right from the beginning. So during the S phase itself, the histone proteins will be produced. Therefore, the correct answer is option B. Now let's move to the next question here. This question was asked in the year 2004. In the somatic cell cycle dash. Okay, let's go one option by option. In G1 phase, DNA content is double the amount of DNA present in the original cell. This is wrong. Okay, DNA replication takes place in the S phase. Yeah, we all agree with that. Option C, the short interphase is followed by a long mitotic phase. Wrong. G2 phase follows the mitotic phase. Wrong. Therefore, the correct answer to this question is option B. You always have to remember that DNA replication or DNA duplication is always restricted to the S phase or of the interphase. G2 phase is followed by a mitotic phase, but here what you have to understand is it is followed by the mitotic phase, which is actually shorter than a interphase. Okay, see, the question is in the somatic life cycle, in the somatic cell cycle, they are talking about the DNA replication. G2 phase actually follows a mitotic phase, but the interphase is shorter when compared to the S phase interphase. Therefore, the correct answer is option B. DNA replication takes place in the S phase. In G2 phase, DNA content is double than the amount present in the original cell. Therefore, it cannot be option D. It is only option B. Therefore, the correct answer is option B. Next question. This question was asked in the year 1996-2000. So, it's a repeated question. In cell cycle, DNA replication takes place in. Very easy question. We already discussed about this in the earlier slide. Whenever we talk about chromosomal DNA replication or simply DNA replication or DNA duplication, we always have to remember it is always restricted to the S phase. What is this S phase? It is also called as synthesis phase. Apart from that, what you can describe about S phase is that they are the important phase in the cell cycle in which the DNA is replicated, occurring between the G1 and G2 phase, right? And this process is a very critical process because you have the exact copy of one is getting replicated, copied or duplicated, whatever you can say. So this phase, if you see, is given of high importance and it is a widely conserved process during the cell cycle. Therefore, the correct answer to this question is option D, S phase. Apart from that, in the solution part, they've explained something more in depth about the S phase. G1 phase is also called as gap 1 phase, is characterized by the increase in cell size. We all know that. In S phase or the synthetic phase, DNA molecules replicate. So where does the replication actually undergoes, or where does the replication process really undergo, is only in the S phase, and it occurs between the G1 and the G2 phase. Therefore, the correct answer to this question is option D, S phase. Now let's move to the next question here. Okay, this is moreover like a calculation. So, but once again, this is easy. You don't have to worry why the question is like this. Okay, a bacterium divides every 35 minutes. If a culture containing 10^5 cells per ml is grown for 175 minutes, what will be the cell concentration per ml after 175 minutes? So for this, I'll show you the solution so that you can understand. So in the solution, you have 175 minutes divided by 35 minutes. Okay, see, we need the answer or the concentration per ml after 175 minutes. Therefore, 175 divided by 35 will give you 5 divisions. Okay, therefore, the number of cells formed is 2. What does 2 represent? See, it is growing in the if the culture containing 10^5 cells per ml is growing for 175 minutes, we have to understand, we have to find out what is the concentration. So 2 to the power of 5 into 10 to the power of 5. Okay, so it is 32 into 10 to the power of 5. Therefore, the correct answer to this question is option C. Okay, apart from that, how can you explain this answer? Any idea? What are we talking about? We are talking about the concentration, right? See, whenever we culture bacterium, we have to remember that we have to remember that they grow in Petri plates. I'm just giving you an extra information. They grow in Petri plates. Okay, you will have a suitable medium inoculated in the, poured into the Petri plates, and the culture will be inoculated now. Now, coming back to the question. So this bacteria, if you see, okay, this bacteria, if you see, each cell, each time the cell divide, you get 2 cells where you had 1. Always you have to remember when it divides. See, if you divide 1, you will get 2 parts. Okay, likewise, each time a cell divide, you get 2 cells where you had 1. You have to remember this, where you had 1. So when we talk about 175 minutes or 35 minutes, the division will always be 5, right? Will always be 5 divisions. So what here is, we have totally 5. That is why we have taken 2 to the power of 5, because we are going to take it as 2 into 2 into 2 into 2. Therefore, 2 to the power of 5. Now you understand how did you get the 2 here? 10 to the power of 5, it is understood, right? Therefore, 2 to the power of 5 into 10 to the power of 5 will give you 32 into 10 to the power of 5. Therefore, the correct answer is option C, 32 into 10 to the power of 5 cells. So after 175 minutes, the concentration per ml will be 32 into 10 to the power of 5 cells. Okay, next question. This question was asked in the year 1997. Which one of the following structures will not be common to mitotic cells of higher plants? Okay. Now we'll go to the solution. Okay, you're given 4 options here: cell plate, centromere, spindle fibers, and centriole. Now, out of the 4 options, it cannot be cell plate. Obviously, it is not cell plate. So it has to be within these 3. So the correct answer goes to centriole. Why? If you see, which one of the following structures will not be common in the mitotic cells, okay, in higher plants? A plant has centromere, a plant has cell plate, a plant has spindle fibers. But plants do not have centrioles. Plant cells do not have centrioles. Cell plate appears during cytokinesis. You have to remember this. When does a cell plate come? During cytokinesis. Since plants do not have centrioles, the correct answer to this question is option B. Okay, so when will the centromere and spindle fiber come now? The centromere will see to that it holds on to the sister chromatids, and the spindle fiber, if you see, they appear during the metaphase. Okay, therefore the correct answer to this question is option B, centriole. Remember that plant cells do not have centrioles. So out of the 4 options, the correct answer is option B. Okay, now to your right side, you can see the centriole. Centrioles are organelles which are only active during cell division. And this centriole is not present in plants, it is present in animals or in eukaryotic cells. They produce spindle fibers which attach to the chromosomes. The fibers pull a copy of each chromosome to opposite sides of the cell, so that the cell, when it splits up, each new daughter cell will have a DNA that it needs. So there is a division, but in that division, both A and B should have an equal division. That is very important, right? Now, when you take, for example, you're cutting an apple for your children, you see to that both your children will get 50-50. So that is the same criteria that is followed here. So option B, centriole, is the right answer for this question. Now, next question. Which one of the following options gives us the right sequence of the events during mitosis? Now, this question was asked in the year 2017. You're given 4 options here. Now look at the first option here: condensation, nuclear membrane, disassembly, arrangement at the equator. So out of this, the correct answer is the first one. Okay, so you're given 4 options here: condensation, nuclear membrane, disassembly, arrangement at the equator, centromere division, segregation, and telophase. So the order, which one follows the correct sequence of order, if you see, it is option A. Okay, So you can see the arrow mark. See, I don't have to explain much about this because the order is very well understood. You only have to follow this order. From condensation, it moves to nuclear membrane. From nuclear membrane, it moves to disassembly. Disassembly, it moves to the arrangement of the equator. And from equator, it moves to the centromere division. And from segregation, it moves to the telophase. So this is the correct sequence out of the sequences given here. Therefore, the correct answer is option A. Now, next question. Anaphase promoting complex. We already discussed this in the earlier parts of cell, the unit of life. So APC stands for anaphase promoting complex, is a protein degradation machinery necessary for proper mitosis of animal cells. Yes, we all agree with that. If APC is defective in a human cell, what will happen? So what is expected to occur? So this question was asked in the year 2017. So what will happen if this machinery of APC goes to malfunction? Okay, see, when we talk about DNA replication or duplication, the major role is played by the chromosome, right? Chromosome plays a major role. So when this APC does not work or does not function properly, the chromosomes will not be segregated correctly. Okay, during the anaphase, okay, this APC is of course happening during the anaphase, is a protein necessary for the separation of the two daughter chromosomes. You always have to remember this in mind. Okay, that is why once this protein APC, this protein is necessary for the separation of daughter chromosomes. So once this APC becomes defective, the chromosomes will not segregate. They will not get separated as two daughter chromosomes. Therefore, the correct answer to this question is option B. Chromosomes will not segregate. Now, moving on to the next question here. This question was asked in the year 2016. Okay, spindle fibers attach onto dash. Okay, easy question. Now, what is spindle fiber? You have to tell me that. What is spindle fiber? What is its function and what is its activity? Why is it a spindle fiber very important for us? See, spindle fibers in cell biology you can describe as, or you can describe or you can refer it to as a cytoskeletal structure of majorly eukaryotic cells, right? Eukaryotic cells. Another thing, it forms, so this spindle fibers, when will you find it? It is only during the cell division. So that is also right. So this forms during the cell division to separate the sister chromatids between the daughter cells. Therefore, spindle fibers, where do they get themselves attached? They attach themselves to the kinetochore of the chromosome. Attachment of the microtubules to the chromosomes is mediated by kinetochore. So what you mean by kinetochore next? I told you what is spindle fiber now. Now you tell me what is kinetochore. Kinetochore can be described as a complex protein. Okay. It can be described as a complex protein. Okay. This is associated with the centromere of the chromosome during cell division. That is why the spindle fibers during the replication or during the process of cell division, cell cycle, gets attached to the kinetochore of the chromosome. So kinetochore is nothing but a complex protein which is required for the spindle fibers to get attached. Therefore, the correct answer is option B. Okay. Now moving on to the next question. Now this question, if you see, is asking us which one of the following is not a characteristic feature during mitosis in somatic cells. Easy question. In fact, this question was asked in the year 2016. So which one is not a characteristic? Okay, we all know it has the presence of spindle fibers. Of course, there is movement of chromosome, disappearance of nucleolus. The only thing which does not happen or which is not a characteristic feature during mitosis is synapses. What do you mean by synapses? Synapses is a pairing of two homologous chromosome. Okay, So that is why it is not a feature during mitotic somatic cell during mitosis. So here if you see, it allows the matching up of the homologous pairs prior to their secretion and possible chromosomal crossover between them. So synapses will take place only during which phase? Prophase. We all know that during prophase 1. So therefore the correct answer to this question is synapses. Which one is not a characteristic? If you see, it is synapses. So that is what they have given in the solution part also. See, synapses take place during the prophase 1 of the zygotine of meiosis. It will only take place through meiosis, not in mitosis. Therefore the correct answer is option D, synapses. So this is the last question for this part 1. Let's read it here. A stage in cell division is shown in the figure. Select the answer which gives the correct identification of the stage with its characteristic. So you're given 4 options here. Okay. Late anaphase, cytokinesis, telophase and telophase. Okay, now coming to the answer. Now we'll go one option by option. Let's read the first option here. Late anaphase. Chromosomes move away from the equatorial plate. Golgi complex not present. No, it's actually wrong. So option A is not the right answer. Option B, cytokinesis. Cell plate is formed. Okay. Mitochondria distributed between the 2 daughter cells. No, cell plate formed we can accept. But the other part of it, what they have given is wrong. Therefore, option B is also not the answer. Option C, endoplasmic reticulum and nucleolus not reformed yet. Telophase is actually wrong. Last option, nuclear envelope reforms, Golgi complex reforms. The correct answer is option D, because that is the correct identification of the stage that they have given in the picture. Therefore, the correct answer is option D, telophase, nuclear envelope reforms, Golgi complex reforms. Okay, while the rest they are talking only about the activity, but they are not telling what the activity or what is it correctly as per the diagram. They have not given the identification. Therefore, the correct answer to this question is option D, telophase. Okay, so with this we complete the part 1, cell cycle and cell division. We will all meet in the next part. Thank you.